We now move to a topic at the heart of modern biology, AI in structural biology. Structural biology is undergoing a paradigm shift where AI enables us to move from static molecular snapshots to dynamic, predictive models of life at the atomic scale. I would like to invite Dr. Gitanjali Yadav from BRIC NIPGR once again to share her insights on the topic.
Thank you, Arhan. This talk was supposed to be given by my colleague, Dr. Devashish Mohanty, but he has not been able to extricate himself from another meeting. So he says he will join us later. What I'm going to do is I'm going to do a very quick, short presentation on what we are doing in the lab and how we are trying to use AI for structural biology. And I'm hoping that, I've shared my slides, so those are not visible. So I'm hoping that with this very short introduction, I will be able to give a kind of a little window into how research laboratories currently, at least at NIPGR and within the BRIC institutions, are using AI. Can everybody see my slides? Because I feel that I'm sharing and it's visible as sharing from my side. So I need to know if others can see my slides. Because I'm somehow able to see my slides, but
I think there's some problem there.
should I connect my computer there? Will it make a difference? Saurabh, kahan pe? Last time when we ran the Unix and HPC course, it was running perfectly, but then my computer was connected to the main device that we have. Let me try again. Can anybody see my slides now? That's what the slides kahan se aayenge? नहीं ये तो keynote है ना पेनड्राइव वगैरह कुछ नहीं है. So that is an issue here. Saurabh, how do I share my slides? या फिर इसे मैं वहां लेके आऊं? देखो, I doubt it. Just give us a sec, please. We're trying to sort this out for the rest of the whole event as well. Okay. HDMI. Yeah. अच्छा फिर, but that's a mirror display.
Yes.
How about in the meeting? It's not going into the meeting. That's the thing. Mm-hmm.
बोल दो ऐसे फायर करेगी तुम यहाँ से करेगी
No, I don't think it is. But can everybody hear me? No, I need to see it on the. I'm not going to trust anybody in this room. Can anyone hear me? I need a text message from somebody out there. I still don't trust because I'm online also and I don't see what they are saying. So I don't trust what I'm seeing or hearing. Okay, so yeah. Thanks. So what I want to say is I wanted to give a short talk about how we use structural biology in the lab, but it looks like our connection is not working. So what we will do is we'll take a 10-minute short break and move on to the next session because we don't want to delay anything. And any kind of delay is basically, keeping things out. If we have time later after the other talks, then maybe either myself or if Dr. Mohanty comes by, then we can give an introduction to the way we do AI in the lab. In the labs in research. Is that okay for everybody? Does that work? Great. In which case, Raghu, are you there? What we will do is we'll keep having tea alongside. Would you like to begin your talk, Raghu? We were planning to do the session from 11 a.m. Would you like to start and try? I'm not sure if Dr. Raghu is here. So we can begin at 11 in case he's not here. Thank you. I see Dr. Raghu in the room. Raghu, can you hear me? Good morning, great. So why don't we, Arhan, let's continue with this. So you'll have to, I think your presentation is better. You can put the main slides just the way you were putting earlier, the speaker slides, and you can continue there. And we'll see how this goes. Yeah, ये नहीं होने वाला यार. Sorry. Next time try करेंगे. Yeah. Over to you, Arhan.
We now begin session 1. I invite Dr. Raghu R to speak on how AI, ML, and rational drug design are reshaping the drug discovery pipeline by reducing timelines and enhancing accuracy. Dr. Raghu Rangaswamy, a seasoned leader with over 25 years of experience in business development and scientific research, spearheads our team of experts. He played a pivotal role in the successful launch of molecular operating environment software, MOE, and Schrödinger software in India, building the business from scratch to millions of dollars. He also conducted user group meetings for more than 1,500 participants, inviting heads for R&D and vice chancellors from universities. His exceptional academic background in pharmacy, biotechnology, and bioinformatics, coupled with an MBA, fuels his unique techno-commercial approach and marketing acumen. Raghu Rangaswamy also held diverse roles as a scientist at DSQ Biotech and Biocon, an assistant professor at PSG Medical College, and a medical representative at Bayer and Tata Pharma. It is my honor to invite Dr. Raghu Rangaswamy to speak.
Uh, Oops. Are you able to see my slides or my desktop?
Raghu, we can hear you, but maybe the timing, the
Are you able to
seconds
see
are
my slides?
not yet. Are you sharing the screen?
Yes, I'm sharing the screen.
Oh, good. Then we can see your slides. So, yeah.
Are you able to see my slides?
Yes, Raghu. Yes, we can see your slides. What about everybody in this room? Are you able to see the slides? Brilliant. Okay. Yes, Raghu, we can.
Okay,
Okay.
thank you.
Brilliant.
Yeah. So thanks, Dr. Geetanjali and the team for giving me an opportunity to talk in this forum. Uh, there is only one thing permanent in this world, that is
Change.
change, right? Probably like, okay, we might have, the people, older people might have seen a phone like this and my generation like this. And today it is something covers every aspect of it. That's what today AI is doing, right? That is covering every discipline in the, not only in life sciences, in every aspect of other areas also. Now, what I'm going to cover today is how generative AI is helpful in the lead identification optimization. How, like, to optimize a lead, it took weeks and months. Today, you can predict within about how the technology is changing. And quantum mechanics, which used to take, if you want to do a DFT calculation for something like 3000 atoms, it takes 2 months. Today, AI does it in just 0.2 seconds. And how one of the toughest challenge is the pharmacokinetics and dynamics, the advent optimization of target prediction. how AI helps in the admit optimization part of it, and also predicting the off-target toxicity prediction, and how AI is helping a lot in the patent analysis, covering the entire world patents, and also in the process R&D synthesis, how AI is revolutionizing part of it, and how one can predict the accurate polymorph prediction, and also in the formulation development and everything. AI plays a lot, but at the same time in the pharma R&D, it is really doing a wonderful job. Let us see on each and everything. So in my opinion, drug discovery is something like a blind man trying to reach an address which doesn't have a door number. That's the same scenario for any drug molecule trying to reach a target, because there are 500 plus kinases in the body and everything is more similar, but on one kinase target. And if you want to reach any place, like, okay, we use a technology like GPS. What is there as a technology like GPS in case of drug? Because we take the molecule here and it has to get absorbed, distributed, metabolized. It should not go and bind to any other potential target. And it should identify and reach its correct target and do its efficacy. It should also have a good retention time, not just like going and reaching the target and staying there for few minutes. At least a molecule should stay in the pocket for a few hours. So this is the challenge in the drug discovery part of it. So how in silico and the AI technologies are helping today? That's what is going to be my talk today. So drug discovery is always assumed as a high risk, a higher time consuming, higher cost a research area. But in today's environment, if AI is being part of it, how it can reduce the timelines, how it can reduce the cost, increase the accuracy is going to be my talk. Now, I don't know how many of you have done molecular modeling or talking kind of things in your research. But if, in my opinion, like in the being in the field for 30 years, we used to take like a huge number of molecules, take so many molecules for preclinical, clinical, and take a longer time to go and file an IND or FDA review or whatever. But today, in the current scenario, one can screen like 100 septillion molecules and filter out based upon various technologies like generative AI, ADMET, binding, unbinding, patentability, off target, everything, and reduce the number of molecules to probably less than 100 or 200. And even today, FDA says no more, reduce the animal experiments, and organoid on chips and other things are coming up, and go with the clinical and reduce the timeline and do your research with a very few number of molecules. So let us see that. And I'm sure like most of us are used to the docking technology, whoever it is. Yes, it's a good technology, no doubt about it. But what a docking technology does, it calculates the various interactions between the protein and ligand, like hydrogen bonding, hydrophobic, philic, pi-pi interactions, pi-cation interactions, various interactions between a protein and ligand. It calculates and then only rank orders, right? It doesn't consider the protein flexibility. It doesn't consider the water thermodynamics, all those things. And even the docking score is not equal to the real delta delta G. It's only a rank ordering tool. Then we started using molecular dynamics, at least to predict the binding affinity, all those things, or the binding mode, etc. But what somebody really needs in the drug discovery research is, and particularly for a chemist, it is like, okay, I'm working on a particular target and I'm going to change some functional group. Let's imagine if you're working on a kinase, you might be changing on the hydrophobic area, solvent exposed area, or a hinge region or whatever. And what a chemist understands is like, okay, the assumption is whatever the thing he is changing, so it is going to give a better binding affinity. That's what, like, okay, is their assumption. But they don't know what would be the real binding affinity that change is going to give. They can only identify after synthesizing the molecule, and that might take about 3 or 4 months, right? But what if a technology can predict as close to the experimental delta G before you synthesize the molecule within the experimental error rate. So you will know whether your molecule is going to come in the -7 or 8, or it is going to be in the -11 or beyond, right? That's what is really needed in case of drug discovery. So you should be able to predict like, okay, the binding affinity of a molecule as quickly as possible. Today with the GPU computing, cloud computing, and all those things, we are capable of predicting these things almost in a day. 5,000 plus molecules, you can predict the binding affinity as close to the experimental binding affinity. So this is really a boon which helps to identify the potential molecules and just take them for the synthesis part of it. But not only like, okay, predicting the binding affinity, today, in my opinion, predicting the binding affinity is not a challenge, right? But what is a challenge, and what is a challenge facing the drug discovery industry all these days is only the pharmacokinetics, dynamics, the toxicity, off-target effect, and all those things. So we all know like a molecule has to have satisfactory things on every aspect of it. It should not have toxicity. It should be easy to synthesize, good physicochemical properties, pharmacodynamics things, good bioavailability, good metabolism profile, everything. Now it is the drug discovery is something like, okay, there are 100 camels are there and the camels to sleep, and you make the 99 to sleep, like, okay, next one will pop up. That is, you optimize one property, the other one will pop up, right? How do you optimize? You cannot go back to the wet lab each and every time. You should be in a position to optimize in the in silico part of it. So that's where like, okay, an ideal drug molecule should be, you should be able to design it perfectly. It should be synthetically feasible. Nobody should have patented it, should have a good SAR profile, should have a for good pharmacokinetics and dynamics, that is how well the molecule is going to be absorbed, distributed, metabolized, excreted, and what is the retention time, and also like, okay, for able to formulate and everything. Today the technology, see, all these days with my experience of 30 years, I've seen like, okay, only like people doing docking dynamics kind of thing. Today, with the AI coming in and with the other in silico technologies, we can optimize all these aspects well in advance using the in silico technologies. And these are the latest technologies in the last 1 year or 2 years kind of thing. So let us see some of the things. And why do you need AI in drug discovery? And how AI is going to be better than a human knowledge or whatever. Let us imagine somebody is coming with an experience of 30 years of experience in pharma industry and everything. So how many molecules they might have synthesized? Probably few hundred, few thousands. That's the knowledge, right? But with AI, one can train millions and billions of compounds. It can be on ADMET, it can be on synthetic feasibility, or it can be on generating the novel R groups, scaffolds, or whatever. AI can give you the suggestions quickly. That's the advantage here. And it's not whatever AI is giving it, you should take it. AI will give you the multiple options. It is a human intelligence. You have to select the best option among them. So let us see like how it is playing a very crucial role. One of the greatest example is like, okay, our the alpha, like, which was able to, we are able to predict the protein structure using the AI part of it. That's a beautiful part of it, and that's where they got the Nobel Prize also. Now, one of the toughest challenge in the DFT, if you want to do a DFT calculation for 3,000 atoms, it used to take 2 months. But today, AI is 20 million times faster, and you can predict the same DFT calculation for 3,000 atoms within 0.3 seconds. Imagine this is going to change the scenario of not only on the drug discovery, even on the materials discovery a lot, because most of the material science people use the DFT calculation a lot. And if something is speeding up, going to speed up 20 million times, imagine how quickly, like, okay, somebody can advance on the research part of it. So that's a beauty, like what AI is contributing nowadays. And there are a lot of AI technologies. We'll come to that one by one shortly in my presentation. So I'm going to cover AI in different areas, right from target identification, high throughput, hit to lead optimization, synthesis, every aspect of it. Even AI is helpful in the even in the NGS part of it. Even if you want to screen whole genome, metagenome, exome analysis, and everything. Now Reliance coming up with like okay, the whole genome analysis with 1,000 rupees. It's all like okay, just the how things have speeded up and how quickly they can analyze. It's like okay, you give a blood sample, like okay, how you get the report tomorrow. So that's the way it is going to change. Now, anybody who is doing a docking, generally people use databases like Asinex, Zinc, or anything kind of things. And conventional docking is like okay, it's only like 2 to 8%. But today, like okay, there are other technologies which is changing a lot. For example, generative AI. Now, if a chemist like okay wants to knows this molecule, let's imagine it is binding at about millimolar concentration. They want to change it to nanomolar or picomolar kind of thing. Now, what a chemist does, they will just go and change the R group, right? But in reality, whatever you are going to change, the change should be within the pocket. If you look into this pocket itself, this ligand binding to the target itself, if you see, this pocket is smaller, this pocket is bigger one, right? And again, when you're changing the R group, okay, it should be synthetically feasible. It should accommodate to the binding pocket and it should bind tightly and it should give a good binding affinity or a good SAR, right? That is the need today for anybody working in the drug discovery. Once like, okay, so here the advantage is the generative AI suggests like, okay, what are the possible R groups possible in this position according to the binding pocket. And either you can quickly screen like, okay, which R group is contributing good binding affinity or even good retention time, everything. Okay, that's where the generative AI comes. That's where like okay, today these technologies are advancing a lot and helping a lot in the drug discovery part of it. Now let us imagine like okay, you have a molecule where the scaffold is patented, but okay, the R groups are free to operate or whatever. Then let us imagine like okay, you want to change only the scaffold part of it. You might be assuming like okay, you might break your head like a chemist has to break the head. Okay, which R group, which scaffold can come here. At the same time, it should react to these interaction points and everything. That's always a challenge. Like you will take time, right? But whereas a generative AI, which is trained on millions of compounds, can suggest which scaffold can fit in perfectly according to the binding pocket. That's the beauty of generative AI. This is a tool developed by Pfizer, and this got many awards, and this is used in their day-to-day drug discovery. So Pfizer and Extalpi collaboratively developed it. So and there is de novo technology. Even if you don't know the molecule, like okay, you can just define the pharmacophoric features and ask the program to design it quickly. Now, the next method I'm going to talk is chemical space docking. And so this is a game changer today. Again, a technology which came last year. Let us imagine you lost your key to your house. Do you go to a person who has thousands of keys and check which key is perfectly fitting? Or you go to a scanner, a computerized scanner, let us imagine, and it analyzes the binding pocket and designs the molecule which are synthetically feasible, purchasable. I'm sure most of you attending the session today are from the biology background. So, and you will all face a problem like, okay, how to synthesize the molecule, right? You will all design, but how to synthesize is a challenge. Now, what the real chemical space is 10 to the power of 60. What we are all used to screen is only like a minuscule amount of it. What we can screen using this chemical space docking is 10 to the power of 30 plus. And in fact, that's what every big pharma does it. That is Sanofi, Pfizer, Evotec, Astra, Merck, GSK. You see, they screen about 10 to the power of almost 30 point of it. How do they do it? Do they have a database of billions, trillions, septillions kind of molecules? No. All they have is the scaffolds, and the reagents. Okay, let us see like, okay, who provides the scaffolds? It is companies like Ambinator, Inamin, Uzi, Watava, Chemspec. There are so many companies. I'm just listing out few companies here who has their own scaffolds. So how the method works is, you might have seen like a child building a Lego toy. So when they want to build a Lego toy, they first put the piece and then look into the protrusion and then see like, okay, what's the next piece they can bring in, right? So the same way, you take your protein structure. Let's imagine this protein structure is having a molecule binding at some millimolar concentration. And you know, like a particular functional group is contributing more for the binding affinity. You keep that program to build it, or you tell, okay, my this one is I don't know what is the ligand here. Okay, completely give an empty pocket, ask the program to design. What it does is, okay, let me see the binding pocket, and it goes and searches the database of the building blocks and brings in a suitable building block, fits it here, make sure like, okay, the next extendable space is here. And once that is done, the different building blocks which suits to the binding pocket are brought. It goes and searches like, okay, what's the next extension? What is the reaction possible here? It goes and searches all the reagents databases. We call it as synthons. If this, under every reagent database, there is hundreds of reagents. And if this reaction can happen, then only that is brought. If that is not going to happen, that is rejected. So let us imagine the next one can react. So the next one is being brought in. So it builds step by step, understanding the pocket and how well the fragment can fit into the pocket. It builds completely. So here, since it starts with like everything novel, every building block is novel, it is not a public domain one. So it builds as per the pocket at every step. So, and again, it not only builds, it also considers the desolvation part of it and everything. It's a super fast methodology, 2 to 3 days, novel unlisted IP, and whatever the molecules are coming out, it is suggesting. Either you can order it and get it from the company who is selling the building block, or you can also synthesize yourself, right? So it works smarter, not harder. And it has a very high hit rate. The methodology, like for example, I'm just going to show two publications. One is in German Journal of Medicinal Chemistry, published by AstraZeneca. This is in Nature thing, Drug Discovery, published by Genentech. So here what these guys did is, there is already a molecule bound in the pocket, and what they did is like, okay, they chopped off a portion of it, a major portion of the molecule, and kept only a small fragment and asked the program to build the rest of it. And they selected only 93 compounds and ordered the 93 compounds. Out of 93, 40 compounds were found to be better than the existing molecule, and some of the molecules were 13,500 fold gain in affinity, and they obtained many crystal structures and everything. So here again 69 compounds. This is completely de novo design, and they ordered only 69. Out of 69, 27 were found to be better binders, several novel chemotypes and everything. In fact, you will be hearing a complete demo of it today by my colleague Dr. Faizan. So same like the chemical space docking, what I explained, the generative AI. Here in the chemical space also, since you have millions of building blocks, analogs and everything, you can just give the structure and say, find out the potential scaffolds which can suit here to this binding pocket, or find out the potential analogs, that is similar molecules or even a diverse molecules which are having a similar pharmacophoric features which can fit into the pocket. Or there is a molecule there, I have a small space available, find out the different potential R groups which can bind to this pocket and rank order them based upon picomolar, nanomolar or whatever. This is what every chemist needs, because they want to find out more options and find among those options, whatever it is giving, which is the best R group, or which is the best scaffold, or which is the best analog, one should be able to identify and proceed in their drug discovery. I'm just curious to know, are you all able to hear whether the slides are like, okay, what I'm talking and everything is coordinating? So just, can somebody tell me? Unmute and tell me.
Yes, all good.
Okay, thank you. So till now, what we saw is like only a hit finding methodology. And I told you like more than 75% of the molecules fail due to the absorption, distribution, metabolism, excretion, and toxicity. And this is a bigger issue because it's like, it's not like, okay, one binding affinity or whatever, because there are more than 200 ADME parameters and a molecule has to satisfy everything. And also there should not, the more issue are on the transporters, the metabolism, the toxicity and everything. And if there is something happening to that, you should be having a knowledge to optimize it, right? And most of the tools offers the solution may have taken. The good thing with AI is, yes, it's fast, it gives you suggestions, everything. But at the same time, the AI also like what is the data you have used to train the molecules. Whether the molecules you have used are only coming from the ADME data from the mouse, then it works only in the mouse, right? If it is coming from a human, then it works in the human. You cannot like use the mouse data in the human. That's where like, okay, today Simulations Plus for partnered with 15 big pharma, took their clinical trial data and generated more than 220 ADMET parameters and also the PK. They are rated as the number one in the world in the ADMET part of it because they are the only one who has the human clinical trial data part of it. They calculate so many physicochemical transporters, metabolism, toxicity, ADMET, high throughput PBPK. If you have your own molecules, you can build the models on the AI models, all cheminformatics and everything. Now, it's not only predicting these property, the other advantage is optimizing these ADME parameters. That is the beauty of this program. Now, if a molecule, whatever you are given, let's imagine you screen 10 trillion molecules and you have taken about a million molecules, like when after screening the 10 trillion, it will say, okay, these are the molecules in picomolar or whatever. You're taking the top 1% out of it. And it's ideal to go and see the molecules which are satisfying all the 220 ADMET parameters. If all the 220 ADMET parameters are satisfied, then it's a green. Now, if something is not satisfied, then only your issue starts, right? Now, the beauty here is it will analyze and say, okay, the log P is more, or the absorption is less, or there is a hug binding. So even any parameter is not up to the limit, it suggests these are contributing only 30% or 40% out of it. So now your other option is, how do I optimize these properties? How do I make like my absorption is only 30%? How do I make it 100%, right? And you are not a DMPK scientist. But here is where the ADMET models comes into play. The AI comes into play. You can just select like, okay, which are the properties are not up to the limit, and you can just click on it and say like, okay, give me the other alternatives, either like change the R groups or change the scaffold or different options, whatever it is, how can I optimize? So it helps you to optimize these ADME parameters also. That's the beauty of the AI today, playing a very crucial role in the lead optimization part of it. Now let us imagine like, okay, it suggested the hug is having an issue. It suggested different scaffolds. Yes, your hug is taken care. Probably even your other parameters are taken care. But what would be the effect on the activity? The activity is reduced or good or better, right? So that's where like, okay, you can use these scaffolds, whatever it suggested, go and do a free energy perturbation. Whether the activity is also good, probably because of your change in some of the scaffolds or R groups, might have also increased the binding affinity or even decreased, right? So because our main thing is, yes, I want to optimize on the ADMET parameter, but at the same time, I don't want to lose my activity part of it, right? So that's where the in silico, the correlation different between different technologies are a must today in the drug discovery research. That's where the AI and the other rational approaches like, okay, mixed kind of thing works together in a synergistic way. Now, I told you like you are all used to docking and we all predict the based upon the docking score. But docking score is only a scoring methodology based upon molecular mechanics. The real thing is the binding affinity calculation. Now, if you want to do a real binding affinity, the delta G binding, generally for a fast method, people use mmGBSA, which takes hours, right? And the R square is around 0.36. But here is a method, a patent pending method developed by Quantify, a Danish company. Up to 1000 atoms in the binding pocket, within 30 seconds, they can predict the binding affinity and with a double the accuracy of mmGBSA. So today we are into a high throughput screening where we screen trillions filtered based upon ADME, now filter based upon the real binding affinity and also go to the unbinding kinetics or retention time. What does this unbinding kinetics or retention time means and how different it is from binding affinity? Binding affinity is how strong the ligand is binding to the target, right? Generally, yes, more the better binding affinity, it might interact and stay in the pocket for a longer time. But the time, the residual time depends from the ligand to ligand. It can be few seconds, few minutes, few hours, even for a day, or even for a longer time, right? So, how long does the ligand bind to the target plays a very important role in drug discovery. The reason is if more the time the molecule is binding, it is highly selective to the target. And the toxicity might be less because it is not going to go and bind to any other potential target. You can reduce the dosage because if a molecule is binding for a day, let us imagine, you can give a 1 day dosage. You can reduce the dosage also, right? So the efficacy is also going to be high. So that's where the retention time prediction is important. The residual time prediction is very important in the drug discovery part. Now, generally, like if you want to predict the residual time, people go for the molecular dynamics, which is a long simulation, takes even days, weeks, and everything, depending upon how much the binding and how much time it takes for the retention time. But here we use, this is again a patented methodology launched about just 4 or 5 months back. We generate a number of unbinding pathways and score the system based upon the activation barrier. So we look into it like, okay, at which side like the molecule is going to unbind and what's the activation barriers, everything. We calculate within a minute or two. In fact, Dr. Vish will talk in detail in the afternoon or after my session. So he will talk more in detail. I'm just quickly running, like explaining you all this concept quickly. So this method works very well on large peptides, kinases, GPCRs, metalloenzymes, all these targets. It works very well. It is like these are the prediction accuracies. It predicts the true negatives, true positives, false negatives, everything. So in our virtual screening, we give importance for the retention time also because so let us imagine in the binding affinity calculation, you are getting like, let us imagine 1000 molecules or 500 molecules within a range of -13 to 14, right? What are you going to select, right? So that's where when you score again on the retention time, you can again sort it out, okay, the molecules which is binding in the binding pocket for a longer time. Now, the other aspects, like, okay, you might have seen many times if you're following the things, the molecule was really good, but it went to the phase 2 clinical trial, it was rejected because of the liver toxicity or neurotoxicity, cardio or nephrotoxicity or whatever. So these things are very difficult to calculate in the human part of it. In the mice, yes, it was able to work because you took a healthy mice and you gave a smaller dose and probably to some extent they might have predicted, but the human biology is different, right? So this is where then Oxford University group has developed like, okay, collected all the 14 million knowledge graph connecting the molecules, the pathways, the adverse outcomes, everything. They've taken the human data and generated a multimodal AI, generative AI and knowledge graph So they can predict like, okay, what are the toxic effects or the off-target effects, what the molecule is going to give. So it minimizes the late stage failures. It helps in accelerating the drug discovery part of it. Let us see with an example. For example, dasatinib. This is used to treat Philadelphia chromosome positive myeloid leukemia. But it also goes and binds to other kinases like BCR, ABL, SRC kinase, and other kinases. Now, if you're working on kinases, you're definitely going to face this problem on binding to other kinases. And if you have the kinase structures, PDB IDs, or even if you don't have, you can just go and use an alpha fold and predict the structures. Come to the free energy perturbation method, where you can analyze on each and every target what is the binding affinity or what is the unbinding kinetics. So this is the experimental delta G and this is the predicted delta G. So on each and every target, what is the binding affinity, that is within plus or minus 1 kilocalorie. If we can predict it, that is really a boon. Let us imagine this is your target, and yes, it binds very well on minus 16 kilocalorie. On other targets, it's about minus 8 or minus 9 kind of thing. And at the same time, let us imagine the retention time is very less, whereas it's really good, then you have hit a jackpot, right? Kind of thing, right? Or even if you're working on drug repurposing, let us imagine your molecule is binding here, but you find out like, okay, molecule is having another target at a good binding affinity. That's again another boon, like, okay, where you can potentially go on target for that. If that is another human relevant target, you can target for that also. Now, AI in patent analysis, since you're all biologists, most of you, like, okay, you might be doing some modification to your sequences, structures, or antibodies, or peptides, or any of those things. If you're working on peptidomimetics or any of those things, and you will not know like whether that sequence, that modification is already patented by somebody. So here is a tool called PateSnap. It is used by 18,000 organizations in the world. So for either for the patent analysis on the sequences or even for chemical structures, you can just give the structure. It will go and report like if somebody has already synthesized, who has synthesized, what stage it is, whether it is in a preclinical or clinical, or for which disease it is targeted. Everything it gives. It searches from millions of databases on like 172 jurisdictions in the world, updated on a daily basis. So it gives you the entire IP intelligence, everything on sequence structures, on even on small molecules, everything. That's where the AI plays a very crucial role here also in the patent part of it. Now, free energy perturbation. This is a method, not a new one, but an old method only. But the beauty nowadays in this free energy perturbation is earlier it used to take months because of the poor computing time, resources, and everything. Today, with the graphics card coming in and the molecular dynamics, we are able to run it these cards and everything. Today, like, okay, it is becoming like, okay, a much faster method. But again, these things depends upon how is your force field and whether you have trained the method using the uh, any known molecules, etc. So this is again a tool in collaboration with Pfizer, where they have done with the DFT calculation and also with the Pfizer's molecule everything. So there are two methods we generally use: relative binding affinity, absolute binding affinity. Relative binding affinity is like this, what I explained. If you're going to change only one portion of the molecule, that is only you are going to change only the hydrophobic and the rest of it is same, or you're going to change only the solvent exposed and the rest of it is, we are going to keep it as such. So that is called as relative binding affinity. On the absolute binding affinity is, let us imagine you isolated a compound from a plant source, right? You don't know, like you're assuming the molecule is going to work on a particular target. You just, it's like same like dock the molecule and see what is the binding affinity for that particular component in that particular in that target. So this again a Pfizer thing. So more than 400 commercial projects they have done. This is a most QM based force field and everything. And today, as I told you, the only thing permanent in this world is change, as I told you in my first slide. So if you are all using docking and taking the molecules based upon the docking score, think of here, it is only an approximate method, as I told you. And molecular dynamics is good on only on protein ligand binding mode prediction, affinity prediction, medium accuracy. Whereas a free energy perturbation is an accurate method, it can be used in multiple instances like non-covalent ligand activity, prediction before and after ligand scaffold hopping, or even an R group change, like I explained you on the hERG binding, protein selectivity, or even on drug resistance prediction. If an amino acid is mutated, okay, because of the mutation, what is the change in the binding affinity happening? Or using that mutation, how can you design something different, like using a generative AI? And what are the other R groups can fit in for that particular pocket because of the like change in the amino acid? Or even for cyclic peptide, bigger molecules like cyclic peptide, covalent inhibition, even if you're working on antigen antibody activity prediction, or even on some protein mutation, whatever I told, everywhere the prediction is really accurate, like probably -7 to 1 kilocalorie, or like, okay, it again depends upon the targets. The accuracy also differs when it comes to free energy perturbation. It is not a magic bullet in every target, depending upon the works on means really effectively. If it is on a GPCRs, it's a challenge. So that it depends upon the targets, whatever you're working on. So here, like, okay, I'll just show you some case studies, but Dr. Bish will explain in detail during his presentation. For example, if you're just changing a proline in the macrocyclic ligand, so it can predict very well. And the other beauty here with Xtalpi is, so they do the generative AI based molecular design, they do the free energy perturbation, they synthesize, they do a wet lab. And if something is not matching as per the prediction, they go back and analyze, like, okay, why there is an outlier. And they again, like, okay, parameterize this part of it and they again make it accurate. That's a beauty here. This method works even on peptidomimetic design, where you can see the correlation very well. Even on cyclic peptides, this we work with Syngene, like where the predicted and the experimental, we were able to predict it really good. And even on a shallow pocket, you can see like within a month, they were able to find out the potential ligands. Even on a shallow pocket like GPX, like they were able to identify 3 potential molecules. This is like generative AI based design, then docking, affinity prediction, diversity selection. So from billion plus to just 30 molecules, out of 30, 10 active molecules, and more than 40% are less than 100 nanomolar. So this is the kind of an accuracy what like today in silico tools are coming in. Now I'll touch upon a little bit on the retrosynthesis on the chemistry part of it. This is another real breakthrough AI is doing in the formulation on the chemistry part, particularly on the process chemistry, retrosynthesis, and everything. So how AI is changing the game, right? This is a publication by American Chemical Society and analyzed by a big pharma. They tested on 9 retrosynthesis software and 60 diverse targets, and they rated the AI works very well. It's not just AI, because if you look into many of the tools available, if anybody is chemistry, you might be using SciFinder, Cynthia, or Reaxis, or any of those things. So these tools works on one principle, either a rule-based or a data-driven, where the molecule is already synthesized and they collect the entire molecule, the entire route, and it is stored in a database. If your molecule is there, whoever might have synthesized all those synthetic route it gives. But whereas here, it predicts based upon all the methodologies, data-driven, rule-based, AI, and even if you have your own data, based upon that, you can train it. And all the expert rule-based, then the CASP method, based upon all these things, it covers 81 million literature and 5 million patent data from across all those things. So you just give the molecule. If it goes and analyzes every step before it suggests a route. Okay, now I'm suggesting a next route. Okay, this is a if the route is coming from the literature, it gives a green color. If it is coming based upon AI, it is a purple color. And if it is coming from a green, all the literature information it gives. If it is a purple, it gives a similar literature or the similar reactions. So this is really a boon in what I'm seeing in India. Like, okay, this is really changing. The AI is really changing because at every step, like, okay, you want to optimize, increase the yield. Yes, you can do it. Or like, okay, I want to reduce the number of steps. AI suggests. Or I want to increase my yield. AI is suggesting that. Or like, how can I reduce my impurity? AI is suggesting. So that is really on the synthesis and also on the process chemistry. This is really a boon. If you want, many times what I have seen here is like where this is really useful is it's not only on the process chemistry and everything, particularly the new drug discovery, because none of them you are trying to design a molecule where nobody has synthesized, right? So when nobody has synthesized, you will not get a synthetic route for that molecule, right? So that's the problem in the new drug discovery part, right? So in that case, like that's where the AI comes into it. Like, okay, you don't have the route. So here is the route, like, okay, based upon the AI, it suggests. So that's the beauty of this method and the AI prediction part of it. AI, and so in all, just don't rely on the AI only. Like if you have AI with another method that works really very well. That's the beauty of this methodology. I have some case studies like, okay, where it has increased the yield, where it has reduced the number of cost, everything. Since you're all biologists and considering the time, I'm just skipping on it. So it covers nearly 600 journals, many patents, 14 patents and everything. Now, you have designed all these things. Now, the polymorphism prediction is another important part in the drug discovery. The reason is like, okay, every molecule will have multiple polymorphic form. Now, let us imagine with all your efforts, you have designed a molecule with a particular polymorphic form, and somebody identifies another polymorphic form which is more stable or like highly absorbable or whatever, then they can go and patent it. Now, how do you identify? Like, okay, any molecule can have multiple polymorphic form. For example, carbamazepine has nearly 27 polymorphic form, right? So when 27 polymorphic form, whether your molecule is like, okay, covered in that, like whether yours is a more stable polymorphic form, you don't know, right? Now, going and predicting all the potential polymorphic form using a wet lab is again a challenging part of it. That's where the AI and the other forms, like it is not only AI, the quantum mechanics and multiple technologies together, it works here. So you have to just give only your 2D structure. It takes all the possible conformation analysis, force field fitting, generates millions of potential possible crystal structures, ranks the energy based upon the clustering and everything, energy landscape. If there is more stable polymorphic form available, it suggests that it predicts almost 98% accurately. And on all free-form, hydrate, salt, co-crystal, even for PROTACs, it predicts almost 100% they predicted. And they worked on nearly 400 plus polymorph projects. And this has got the award for the last 8 years consequently in the Cambridge Crystallography Data Center blind test. Like you all might have heard about the CASP test in protein structure prediction. So like that, here is a blind test for the crystal structure prediction, where they give the toughest targets, the molecules having even metals like silicon, copper, or even materials, everything. So in all these things, like it has predicted even up to Z prime 2, it predicted every polymorphic form accurately. Now, formulation is another important part in the drug discovery. You all might be observing like a molecule is coming out of a patent. Okay, then like, okay, the challenge for an Indian generic company is how to formulate, right? What, because they will not define like, okay, what is the ingredients they have put in, right? So here, like, okay, you have to design like, okay, this if an API is coming out of a patent, you have to see like, okay, for this API, which excipient is going to be suitable and what is the combination of the excipient you have to add. And by adding that combination, there is no impurities are generated. The stability is good. The drug excipient compatibility is good. And this combination, like what you are making, it matters a lot in the stability of the the formulation, whatever you are doing. Because let us imagine you're doing a syrup or a suspension, it should not sediment and other things. So this combination has to be optimum, and your API is soluble in which solvent, all those things, right? So there are a whole lot of things in the formulation study also: drug-drug compatibility, drug excipient, formulation stability, impurity control, permeation if you're working on skin, packaging materials also, like there is a challenge, solvent selection, all those things. So here again, or even the solubility prediction part of it, we predict on 11,000 solvents in 8 hours, about 85% accurately. So this is really a boon for somebody working in the formulation research also. Now, whatever I explained, you can combine all these tools, and particularly most of you might be structural biologists. You can bring in all your collaborator. They can be any part of the world, and whatever the modification, let us imagine a generative AI is suggesting a free energy perturbation, you have done it. You can walk, sit into the protein, analyze, okay, what interaction it is making, or what hydrogen bonding or electrostatic interactions it is making, or what are the ADME parameters. You are all apart in the world distantly, somebody in US, somebody in UK, somebody in Germany, or whatever, but it gives a feeling everybody is walking into the room and designing a molecule collaboratively. This is used by all the multinational companies in the world. If anybody is a structural biologist, you will really love it. Now, LLMs. So we are all using the probably chat GPT in a day-to-day kind of a thing. I'll tell you like how this is really changing the world in the pharma world. Let us imagine you have a lot of database sources, ComChem tools, context info tools, bioinformatics tools, or like any of the World Wide Web like PDB or whatever, or even PubChem, anything. You can just give a command like how you're doing in Alexa, dock this molecule to this PDB ID and show me the results. That's it. You don't have to do anything. It goes, take the structure from the PDB, takes the ComChem tool, whatever you have, does the docking, everything, and sends you the results into the thing. The other beauty here is you can also just like draw the molecule and say like, okay, calculate me the ADME parameters. So if you already have a tool locally, which the API is connected, yes, it can go and calculate the whatever the molecular weight or ADME parameter or whatever you want to ask the details from any of those tools. Like you can take it, or you can say, does this molecule have a crystal structure in the PDB, right? Because many times what I hear every day is when I give a talk, like, okay, I have a molecule, I don't know which target it is binding, or what are the other potential targets it can go and bind. So can you find out like, okay, what are the similar molecules of this in the PDB, right? So just give a command, it goes, searches the entire PDB, like, okay, how you're getting the results from the chat GPT. It goes and says, these are the PDB IDs, which are similar to your molecule, and brings those PDB structures and shows that even if you want to write a code, don't break your head to write a Python code. It writes the Python code like for you. So you can search like, okay, all the even if you want to like, okay, most of you might be the bioinformatics and kind of people here. So you might be doing day in and out like sequence analysis, structural analysis, and all those things. You can do all those things. You just give your sequence to this. Don't go to like, okay, which tool I have to take and everything. It just does the sequence analysis, structural superposition, okay, the protein sequence analysis, from all those things it takes and does it and gives you the results. So let's imagine like, okay, you're doing a structural analysis, you want to superimpose all those structures and see like, okay, because you have fetched out the similar ligands from the PDB, and probably you wanted to see like, okay, in each and every target how it is binding, or like what would be the off target effect for your molecule. Let us imagine like, okay, in the bios or the biotox, I told you like what are the off target effects it can have. So you can see like, okay, whether it is there is any similar ligand in the other target, whatever it is suggesting, what are the potential, somebody has already crystallized everything. You don't have to go and use multiple tools. One tool, you can go and do all your research, get the details, research information, everything. Even if you want to do a statistical analysis, or you want to do a graph, or compare different properties, you can say plot all these things, like, okay, it does the plotting also. So it is a combination of multiple tools and the World Wide Web. Now I'll come to a next one. This is really an impressive one. This is what every pharma world is going ahead. Now you are doing let's imagine a pharma company, or let's imagine a process company, like, okay, process chemistry company doing some research, or a new drug discovery company. You might be doing in silico research. You might be doing getting the data from all the wet lab experiments, probably 1,000 instruments. You might be getting the data from NMR, X-ray, ELISA, qPCR, NGS, and everything, or like all the experimental metadata, all the scientific content, whatever you have. And you might be also having a factory where, like, okay, that might be connected, and there different kind of equipment, everything comes in. All the data are collated. You can do a generative AI, or you can do an LLM. You can ask any question. Okay, now you have my in silico data, you have my wet lab data, instrument data, everything. Okay, suggest me like what way I can optimize. That's where the technology is changing, like where integrating all the data, analyze them, everything. So none of you are using the black and white TV or a mobile phone like this, but probably the technology is just 2D, 3D QSAR were the predecessors for today's AI/ML, no doubt about it. Talking and dynamics were the predecessors were today's free energy perturbation and retention time. But today the technology is changed. We are all using these technologies. It is time, because if you want to identify the potential molecules, then, like, okay, potential R groups, scaffolds, or analogs, generative AI gives you the options quickly in seconds. You don't have to break your head, and it gives you the multiple options. Just do a docking, or just do a free energy perturbation, which R group is contributing more for the binding affinity, or you're just starting from scratch, or you want to take a millimolar molecule and change it to micromolar or a picomolar, use a chemical space docking using 10 trillion or septillion kind of a thing. If you have your own building blocks, bring that. ADME today is not an issue. Based upon the clinical trial, you can predict the ADME data, you can optimize the ADME data, you can also optimize the toxicity, off target effects, predict the quick binding affinity, unbinding kinetics, and check whether the molecule is patented, predict the accurate binding affinity, use a generative AI based synthetic route prediction, and formulation design, everything. And I would say, like, today, like, okay, chat GPT, everything is free for us, right? Because they take the data. But we are all doing our hardcore research. These should be stored in the electronic notebooks. So now prepare yourself for the future of innovations. In IT, we are already seeing AI is replacing the humans in writing the codes. I read in a newspaper article, like, where the last year only net hire is only 17 people, right? But in drug discovery, AI, ML, and cloud computing will be an amplifier. It is reducing the time frame from 15 years, reducing the cost, helping us to optimize a lot. That's what I would say. But it's not going to replace humans. But humans who know AI will replace the humans who don't know AI. And my suggestion and request is, just don't blindly rely on whatever the AI is suggesting. AI is giving you multiple options. These are the R groups, or these are the synthetic routes. Now you have to use your knowledge, your intelligence, and select what is the best, and use the multiple technologies, cross check with that, and go for your research. That is going to accelerate your research drug discovery part of it. We are all used to all these technologies we use day in and out, and we are also evolving a lot. It's time for a paradigm shift in our drug discovery research also, right? That's where, like, okay, we are in an era where the AML technology is well matured, I would say. Not, but, like, see, in drug discovery, it again depends upon the amount of data, where the data is coming from, how well the data is curated, whether the data, like if you take an ADMET and other things, whether the data is coming from the human clinical trial data, that all matters a lot. But at the same time, today we are in an era of GPU computing, cloud computing, and now the quantum computing is coming in. In fact, like, okay, I just wanted to inform you, like, okay, I didn't cover on this quantum computing part of it, but if somebody is interested in the quantum computing based binding, unbinding prediction, there is a tool, the Quantify, whom we have partnered with. They are conducting a hands-on session today. If you want to participate, I'll send the link to Dr. Geetanjali. You can register it and attend that. That is really a wonderful session. And it's time to adapt to all these available technologies. Multiple disciplinary coordinator research is the mantra today. In fact, like, why we have partnered with all these companies, because you have seen, like, you might have seen, like, okay, even in my presentation, so I'm screening from millions of trillions of compounds, filter out by ADME, filter based upon quick binding, unbinding. Then you go and predict, like, okay, the free energy perturbation, predict, like, okay, patented. No company provides all these tools in one company cannot provide everything, right? That's where, like, okay, we partnered with multiple companies where we bring in the technologies. And if you are involved in the drug discovery, it's a multidisciplinary coordinated, and you have to use these multiple tools and technologies. And we feel like, okay, we are in the right time to implement AI into our R&D. India excels a lot in the space tech, but it's time, like, okay, we should also enter into this, and we should also think of, like, okay, we as a country, we should not just keep doing services. We should shift from service to innovation. We are also establishing Center for AML, Center of Excellence in Drug Design. Last week, I established this one with the MIT ADT University. So we are trying to bring in all the tools in one place, and whoever wants to establish, we are establishing it. Thanks a lot for giving me the opportunity. And I'll stop here, and probably you can unmute and ask the questions. I'm ready to answer the questions.
Thank you for such an informative presentation, Dr. Raghu. Some of our participants have written down some questions in the chat for you. If you can just take a few minutes to go through those, and then we'll
okay.
go on to the next session. Thank you.
Wait a minute. I'll just go to the chat portion of it. Okay. Because when I'm presenting, I'm not seeing the chat. Let me see from first. If anybody has, okay. That means AI generated the exact root structure for further use. Yes. So AI generates the exact structure for further use because it gives you the possible potential R groups it gives. How could we validate the accuracy of AI while generating the root structure for further use? So if you are working in the new molecule, then, like, okay, you have to go and do a free energy perturbation and other things. Always, whatever the in silico, it is a suggestion. It is telling, like, okay, these are the molecules which will have a good potential, or in case of synthesis, these are the potential routes. And now when it comes to it's a good question, like, okay, when it comes to the AI part of it, what we have observed is, so it generally gives, like, okay, we can search based upon, like, okay, I want a accuracy, like, okay, the risk, lower risk, medium risk, high risk. When you say high risk, then what it suggests is it gives you more AI based routes. And when it gives, like, generally it gives, like, with the literature mixed with AI, both the options it gives. But in some of the challenges, what we have seen is it gives, like, okay, if there is nothing available completely novel, it gives completely an AI based thing. And we have observed up to 7 steps it is accurate, reproducible. Beyond 7 steps, it is a questionable. So whatever it comes, whatever the routes it suggests, like, definitely you have to revalidate. And in the drug discovery part, you cannot replace the wet lab. Understand that these are all suggestions which is giving you, like, 80% success rate, you are going to succeed. So rather than, like, okay, if we, in case of binding affinity, rather than -7, I'm going to select the -12 or 30, right? So I'm picking up the most probable successful candidates. That's what the AI in silico, everything helps. So that's the wet lab is the correct validity. Can we add 2 more protein sequences to generate 2 or more protein sequences generate engineering part? Yes, like, you can do that. Are these tools available for free? Sorry, like, they are not available for free. They are all chargeable, but you are getting these things at about 1 to 2% of the commercial price. In fact, the admit we give it free. It is about 2 crore per commercial usage, but like you get it free if you are having the Center of AML drug discovery. Diseases have a large spectrum of presentation depending upon genetic heterogeneity as well as molecular interactions of these variants with other molecules. Of course, we have to start at the core of the disease, which is a faulty protein or a toxin or a gene. But how do you suggest we can capture the larger picture from non-target effects on the drug side effects? So that's where like the biotox, which I was explaining, was on the other non-target effect. So it covers all the potential other targets. And again, like, okay, if you're giving a molecule, the MARA, which I told you, that covers like, okay, what are the other similar targets, like, okay, what the molecule is binding. See, unless we know the entire PDB structures or the entire molecules, like, okay, we can only compare to another similar one or whatever, and then like, okay, what's the other off target it can happen. Hoping to see more integration of LLMs and Unreal Engine AR in medical and clinical applications in India. Yes, interesting term. How can we do molecular dynamics simulations using AI, sir? Which tool is accurate, sir? Okay, so we do have a tool on the molecular dynamics, but I didn't cover on that. But on the quantum mechanics part, I covered like, okay, where like it is 20 million times faster on the quantum mechanics part. But molecular dynamics is still like, okay, a tool has to be done using the real. I don't know like how far the AI can replace on the molecular dynamics. Is any software which can be used to check, predict active, inactive peptide ligands? Yes, free energy can be used. In fact, like there is a CSAR where like, okay, if you are having a binding pocket, a protein, we have a grow mode. And I'll ask like Phison or whenever you're taking, this is a more of a biology group. So cover them on the peptide design also. So you can also like, we have a tool like the grow method, like in the binding pocket, how you can add the fragments and design. The program automatically adds what is the next fragment, what you can add. Same way, like on the peptide design, also you can add it. And I'll ask Faisal to cover that in his demo part of it. Is training data and source code available to build our own such models, to develop such tools, and what might be computational requirement? Okay, we have a tool like the Yasara, like which you can you can change the things, but not with other tools. With the rise of AI, GPU computing and cloud computing are the next cloud computing are the next big thing. As like Nvidia, Oracle, like other companies are rising with respect to drug discoveries and healthcare. With the rise of AI, GPU computing and cloud computing are the next big thing. Yes, this is playing a very crucial role. GPU, cloud computing are playing a very crucial role. At the same time, next the quantum computing is going to play a very crucial role. Today with the kind of NVIDIA chips coming in, like, okay, if you're all working on and using molecular dynamics and other things, you see like every time, like, okay, in fact, that is the fastest changing technology, I can say. I have seen from the days of K20 and all those things, and today like H100, A100 and new things coming up. And the chip cost also, and you know, the most valuable company in the world is NVIDIA. These chips are changing really a lot, kind of particularly the computing power, and the cloud computing is something really another wonderful thing. In fact, like in Delhi itself, there is a company who uses 1,000 GPUs things and use the free energy perturbation. And there are other companies who are using 1 lakh GPUs and do the free energy perturbation calculation, because the free energy perturbation calculation is the most accurate binding FD. Now with this, uh, the other quantify which is coming in, which predicts within a minute, that is also changing. So if target information is unknown, if we have to start from molecule only, how would we score unknown biological activity of that particular molecule? Can we use generate AI for this purpose? Okay, if the target information is not known, like, okay, if you are having only the molecule, there are two ways. One, you have to compare with the known existing drug database, because like, okay, this molecule is similar to what are the other similar molecules. You do a similarity search, or use the Mara, which is just about $300 for academics. So in the Mara, like, okay, you can just give the structure and you can say like, okay, what are the other similar structures in the ChemPDB or in the PDB, ChemWell or whatever. So if somebody has already synthesized the molecule and that information is there, it will fetch from the entire World Wide Web and give you the information. So that's the way, like, okay, you can do this, because when you don't know, like, because there are 30,000 structures are there, like we don't know the PDB structures for everything, right? Either you have to go and talk to everything. The other easiest way is, this is my molecule, these are my pharmacophoric features. What are the similar pharmacophoric features and what are the targets it is acting on? And then, like, okay, you take that, you do an alpha fold structure prediction and do a docking or free energy perturbation and predict, okay, how well it is interacting in that target. That's what is the kind of technologies it's available today. So if there is good, like, a lot of questions, I'm sorry, like, okay, I have to do it only virtually, and I know, like, it's a short arrangement, but anyone, like, okay, feel free to contact us if you want to evaluate the software on your research part of it. You can uh, send a mail to my team or to me, and we are there to help you. We have brought in the best technologies, whatever is there. Like, we are hunting, like, okay, the good latest technologies, and we wanted to bring it to India. That's our aim. So you will find it, like, okay, many of these things, like, okay, whatever we have, we evaluate, and if it really works, then only we partner with them. So that's where, like, okay, as I told you, like, okay, change is only permanent, and you have to adapt to whatever is the new technologies. And particularly when you use any AI tool, make sure, like, okay, don't use it as a black box, where how they are getting the data and how much of a data they have. Ask those questions and then use it in the research. And it's always better, instead of going with a pure AI with other rational methods, if it is there, because that will validate and, like, okay, given kind of a more accurate prediction. Because we have also analyzed many pure AI based solutions which they claim tall claims, like, okay, we predict very well these, that, everything, and many cases it doesn't work. So that's the kind of thing is our observation. Okay, if there
Thank
is any questions, like, let me know. Otherwise, like, Dr. Geetanjali, like, okay, I don't know who is the next speaker.
Yes, thank you, Dr. Raghu, for such an insightful session one. If nobody else has any questions for the kind doctor, we will be moving forward to the next session, session two, in just a minute.
Thank you. Thanks a lot for the organizers for giving me an opportunity. I hope, like, the participants also gain some information on this new technologies, AI and everything. Thank you.